Hello, and welcome. I would like to share the results of my master degree dissertation. The title of my work is Deep Learning Neural Network Image Analysis of Immunohistochemical Protein Expression Reveals a Significantly Reduced Expression of biglycan in Breast Cancer. In women, breast cancer is the most commonly diagnosed cancer. This is the leading cause of death from cancer in the world. Breast cancers are heterogeneous in nature, both at the histological and molecular levels. The molecular profiling of breast cancer are important because it guides diagnostic and therapeutic strategies for this disease. New breast cancer biomarkers have been sought in order to better characterize the tumor and to select the best possible treatment. Biglycan is a class 1 small leucine-rich proteoglycans. Its gene has been mapped to the chromosome 10. Biglycan is characterized by a protein core with leucine-rich repeats and is composed of 331 amino acids. When fully glycosylated, the molecular weight of biglycan up to 250 kilodalton due to the presence of 2-chondroitin, a dermatan sulfate, and glycosaminoglycan chains, covalently attached to the N-terminal region. Biglycan may alter tumor proliferation by modulating the tumor microenvironment. However, the clinical impact of biglycan on cancers is still poorly understood and sometimes contradictory. The type of antibodies used in immunohistochemistry and the specific site for biglycan identification may explain these discrepancies. The monoclonal antibody from Abnova is against the full sequence of biglycan. Data on biglycan expression in human breast cancer is scant and contradictory, and little information is available using the Abnova antibody in pathology sections. Therefore, the objective of this study is to verify the immunohistochemical expression of biglycan in breast cancer biopsies, compared to normal breast tissue. We will use a validated monoclonal antibody and 2 digital imaging methods of analysis, digital H-score, and deep learning neural network image analysis. This is a case control study. Paraffin blocks, dated between January 1, 2012, and December 30, 2015, were obtained for analysis from the Pathological Archive of Hospital de Clínicas in Porto Alegre. Slides with benign and cancerous breast tissue were reviewed by a certified board pathologist to confirm the diagnoses. Women aged between 20 and 79 years old with diagnoses of invasive ductal carcinoma, and those who underwent breast surgery for benign conditions, were included in the study. Patients with lobular carcinoma, or intraductal papilloma, who had undergone chemotherapy or radiotherapy were excluded. Immunohistochemistry was performed from paraffin sections 3 micrometer thick. Primary antibody against the full length of biglycan was from Abnova. The primary antibody was visually detected using diaminobenzidine as chromogen and counterstained with hematoxylin. Negative controls were obtained by replacing the primary antibody with isotype control. Human lung cancer samples were used as external positive control. Photomicrographs were coded and blindly analyzed. Image analysis was performed using digital H-score in ImageJ software. Only the glandular and tumor sites of the tissue sections were selected as regions of interest. After selecting the region of interest, images were submitted for color deconvolution analysis. The intensity of the DAB was calculated by ImageJ, reducing subjectivity. Another set of DAB-only images, with 100 times magnification, and without marking the region of interest, was submitted to the Inception v3, Deep Neural Network Image Embedding Recognition Model Analysis, using Orange 3 software. The sample size for ImageJ analysis was calculated in order to have a power of 90%, an alpha error of 5%, and a standard deviation of 20, to identify a decrease from the average of 40 DAB units in normal breast tissue to 4 DAB units in cancer. With these figures, at least 7 samples in each group were necessary. Sample size for supervised neural network analysis was chosen for convenience after obtaining the maximum number of photomicrographs from the slides. Fisher's exact test was used to compare categorical data. Unpaired student t-test with Welch's correction was used to compare the expression of biglycan through DAB intensity between the two groups, if data had a Gaussian distribution and different standard deviation. Gaussian distribution was calculated with the D'Agostino and Pearson omnibus normality test. If Gaussian distribution was not identified, Mann-Whitney U-test was used instead. Supervised neural network analysis was performed using stratified 20-fold cross-validation with 200 hidden layers, ReLU activation, and regularization at alpha with 0.0001. This deep machine learning process classifies images into two groups, cancer or no cancer, without any human subjectivity. Ethical approval was obtained from local ethical review board and registered at the Brazilian Research Platform. From 24 potentially eligible cases, 15 cancerous cases and 9 benign cases were obtained for analysis. The mean age and the ethnicity between groups were not significant. The mean biglycan protein expression, in DAB units, was significantly lower in breast tissue with cancer compared to benign breast tissue. Panel A depicts biglycan protein expression in invasive ductal carcinoma, while Panel B is a representative expression of biglycan in normal breast tissue. External negative and positive control, as shown in Panel C and D, respectively. A total of 129 high-power magnification photomicrographs with DAB staining-only images were submitted for supervised neural network analysis. The analysis yielded an accuracy of 85.3%, demonstrating that it was possible to classify samples into cancerous or benign tissue using only the expression of biglycan. In conclusion, biglycan protein expression is reduced in breast cancer tissue compared to normal tissue using a specific monoclonal antibody. These results were confirmed using two different techniques with little or no human subjectivity. Thank you for watching.